All right. Secondary neurulation, or as our title slide says, secondary neurulation. If you ever do these chat GPT images or AI images, like they really have a hard time with images of like text or whatever. Emonotion. But this looks good. Like some of the stuff actually looks okay. Anyways, let's go. So, secondary neurulation is in the caudal region, and this is in a chick embryo. So remember, caudal is toward the back. In this case, we're in a 25 somate chick embryo. Again, this might not mean much to you right now. We'll talk about somates going forward. The mesenchymal cells, they condense to form a medullary cord, as they call it, at the most caudal end of the chick tailbud, so towards the very end. You can see here that it's starting to form. And this is, remember, instead of the folds coming in, this is where the mesenchyme kind of melts into these neural cells. So we have that here. We have a transitional region where they're turning into the medullary cord. Then we have this cavity formation, right? The neural tube is cavitating, and the notochord starts to form. And you can see there's two like separate lumens here, interestingly, where it's not just one cavity, but then eventually they merge together. These lumens coalesce and form a central canal of the neural tube. So remember, this is different than the primary neurulation where we have just the folding. And so here they've done, this is kind of GFP stained, right? So we have blue, blue is DAPI, so the nuclei. Generally, whenever you see blue in these fluorescent images, it's DAPI, so it stains nuclei. And then the GFP here is, let's see, superficial cells of the primitive streak are labeled with a cell permanent fluorescent green dye. So you can see here that these cells are turning, are starting to kind of coalesce down here. The neural tube is going to start to form. Look at the difference in time too. It takes quite a bit of time, 15 hours, to get from these coalescing cells down here to this neural tube formation. At 3 hours, the superficial cells can be seen as starting to ingress. So you can see they're moving in from this neural plate. You can see a few examples here. And then these will start to, the mesenchyme will start to transition as well. And then this yields the neural tube at 18 hours, roughly 18 hours, depending on the organism and such, give or take. So also, we're going to, in talking about this neurulation, so we're talking about the formation of the primary, so like your hindbrain, and your secondary, so down lower towards your lower back. But we also need to talk a little bit, excuse me, I'm boring myself. We want to talk about early brain development in this lecture, because otherwise this video would have been 3 minutes long. So we're going to loop in some early brain development. So here we have, this happens at the most anterior, obviously. Up we have the neural plate, and then we have the notochord down here and the primitive streak. So neural plate, primitive streak regressing here, the notochord being laid down. We have formation of this neural groove. And so, let's see. The neural fold begins closing at the dorsalmost region, so up towards your back the most, forming this neural tube. Let's move on to this part. So here, you can see up at the very most anterior part, we have this neural tube forming, but we also have this brain starting to go to develop here. So the neural tube develops, I couldn't find the word, develops these three brain regions and the spinal cord. So you have the spinal cord, which is a little more posterior, And then we have the hindbrain, the midbrain, and the forebrain, all at the most anterior part of the neural tube. The neural tube remains open at the most anterior portion. So at the very, very top of your forebrain, it remains open. And you get the optic bulges. So those, let's see, do we have those? They're not, those are these, will eventually develop into the retina. Let's move on to the next slide here. Slide E. So in humans, the three primary brain vesicles become further subdivided since we have more evolved brains, right? And development continues. So you see that we have our primary vesicles, forebrain, midbrain, hindbrain, but then we start getting additional subdivisions. So we have the telencephalon, the diencephalon, the mesencephalon, the metencephalon, the myelencephalon, and these all develop, if you've taken psychology or anything that talks about the lobes of the brain and what they do, or maybe neurobiology, I don't know. But you have the olfactory lobes, right? So that's for smelling. Your hippocampus, cerebrum. So we have memory, storage, and intelligence. Then in a little bit further back in the hindbrain, we get the optic vesicle. So that's for your vision, where your retina develops from. Epithalamus, which is your pineal gland, your thalamus, and the hypothalamus. So those are kind of like your temperature, sleep, a little homeostasis type stuff. But your midbrain also has some of that as well, right? So with temperature regulation, cerebellum. I'm not going to read all of these. Medulla, and so your hindbrain, they always say, is like your ancient brain. So it's like the not as evolved, very basic functions, I guess. So we did this in one of our case studies, the rhombomeres, and these are just the kind of help lead to the development of these lobes of the brain. So the roof plate has been removed so that you can see kind of the inside. This is like if you're looking, take the first layer off of the developing brain. So in part B here, they've just stained this with a antibody to kind of show these neurofilaments. So you can see that we're starting to get kind of like these neurons that are developing in these rhombomeres. And these rhombomer boundaries, so kind of like these dark spots in between them, these are emphasized because they serve as channels for neurons crossing from one side of the brain to the other. So those early in development here, we have kind of our connected portion of the brain where these neurons are moving, are connected from one side to another before the lobing really starts to take off. But yeah, so anyways, just thought it would be interesting to show you kind of the sustained development of these neurons as they're developing in this early brain stage. That is it for this. I broke off this video from a video that we're going to do some like anterior-posterior, dorsal ventral accessing in the developing spinal cord and neural tube, because that's very important too. So if you know much about spinal injury or people that have had spinal injuries or pinched discs and things like that, where your nerves connect to your spinal cord, whether it's more dorsal or ventral, those all affect different parts of your body, and like where your nerves come out. So like I'm getting old, and I had a pinched disc in my lumbar region, and I made a joke. I was teaching genetics at the time, and I told my class, I said, I feel like I'm having like the world's longest heart attack because like I had like tingling in my left arm for like weeks. They say that's a sign of a heart attack. But I had a pinched disc, and the disc that was pinched, those nerves just controlled like this left side of my body. And it was only like these two fingers, which is interesting. So anyways, all that being said, is a long way for me to say the anterior posterior axis and like the real, like all the coordinates of the developing spinal cord is really important in that pattern formation, because they all control different parts of your body. So we're going to get into that for kind of our last part of the neurulation talk in the next video. So thanks for watching. I'm going to keep this short, otherwise you guys know I'll ramble forever. But I will catch you in the next video.